Welcome back to the deep dive. So, you probably think that, modern medicine, we can see everything we need to see to diagnose diseases, right? We look at where proteins are, kind of their general levels.
Right, the big picture stuff.
Exactly. But what if the real action, the moment a cell actually flips that switch towards being diseased, what if that's happening on a scale we just can't normally see, like tiny shifts inside the cell parts?
That's precisely the blind spot, that invisible action. It really limits what traditional pathology can tell us. Even the fancy multiplexing tools we have now, things like IMC or Codex, they're great for telling you what cell it is,
Okay.
but they often top out at maybe 30, 40 markers.
hmm
And the resolution, well, it averages around 267 nanometers. Good for identifying the cell type, but not quite sharp enough to see that detailed molecular dance happening within the nucleus or mitochondria.
Right, you missed the critical subcellular details. So, okay, we need a finer lens, and you've brought us something new today, a framework called pathology-oriented multiplexing, or PathoPlex. Sounds like it's designed for exactly this.
It is, yeah. PathoPlex is built to overcome that resolution and integration barrier. It's basically a new workflow combining really high-res imaging, subcellular level, with some clever software.
Okay.
And the key output, what it's really looking for, are these protein co-expression patterns. They call them clusters. Think of a cluster as a biological signature. It bundles together information about the cell's structure, where proteins are inside the cell domains, and even signaling activity all at once.
So it's not just more detail, it's integrated detail. The breakthrough isn't just seeing 100 proteins instead of 30.
Exactly.
It's that PathoPlex defines disease based on this nanometer scale fingerprint, this cluster, that signals trouble before maybe the whole cell looks obviously sick to standard methods. What kind of scale are we talking here? How many markers? How sharp is the focus?
It's a pretty significant leap. PathoPlex has been optimized to map, get this, over 140 different commercial antibodies.
Oh, 140.
Yeah, across 95 separate imaging cycles. And the resolution is down to 80 nanometers per pixel, which is incredibly sharp.
80 nanometers.
And crucially, they tackled the logistics. They figured out how to process at least 40 archival biopsy samples. We're talking old, formalin-fixed, paraffin-embedded tissue, FFPE, the stuff hospitals have stored for years, process them all in parallel.
That's huge for actual clinical research. So today's deep dive, we're going to unpack the technical magic behind this jump in scale and detail.
Right.
And then look at the really cool part, the discoveries it's enabling, especially in tough areas like kidney disease.
Exactly. It's about shifting pathology from just classifying cells to, well, reading these subtle spatial molecular signatures of disease. It's data-driven pathology.
Okay, let's dig into the how. Going from maybe 30-ish markers to over 140 on delicate old tissue, cycling 95 times, that sounds incredibly difficult to pull off reliably. What are the core innovations making this work?
There are really two main pillars here. First is the imaging framework itself. They call it 4I, which stands for iterative indirect immunofluorescence. And second is the software engine that makes sense of all the data, called spatiomic.
Let's start with 4I. If you're staining and imaging 95 times, how do you avoid signal bleed-through? How do you make sure the signal from cycle 10 doesn't mess up cycle 11? And how do you keep the tissue from just disintegrating or floating away?
Yeah, those are the key practical hurdles. The signal cleaning part relies heavily on the elution step. After each round of imaging, they wash the tissue with a pretty specialized chemical buffer.
Sounds intense. What's in it?
It's a mix of things like glycine, urea, guanidine hydrochloride, and TCEP. The whole thing is adjusted to a very acidic pH, around 2.5.
Whoa, okay. That sounds like it could dissolve anything. How does it clean without destroying the tissue?
It's carefully optimized. Think of it like a very effective but gentle molecular scrub brush. It's designed to strip off the antibodies from the previous cycle completely, but without messing up the underlying tissue structure. And they showed it works. The tissue structure stayed intact through all 95 cycles. The elution is really, really critical.
Okay, so the signal is clean, but keeping the tissue itself physically stuck to the glass slide for 95 washes and chemical treatments, that seems like another major challenge, especially with fragile biopsy samples.
It absolutely is. And this is where they made a really important practical discovery. Most labs use something called poly-D-lysine to coat slides and make tissue stick.
Right, standard stuff.
But PathoPlex testing showed that even with polyglycine, about 15% of their kidney samples started lifting off the slide after just 49 cycles. That's obviously not workable for large experiments.
Yeah, you lose data, waste time.
Exactly. So their solution was switching to a different coating, APT, which is 3-aminopropyl trithoxysilane. With AP's coding, tissue lifting dropped dramatically, down to only 1% in kidney samples. Sounds like a small detail, this adhesive chemistry.
But it unlocks the whole process at scale.
Precisely. It's one of those enabling, unsung hero kind of findings.
Okay, tissue anchored. Now, alignment. You imaged the same spot 95 times. How do you ensure pixel number 5 million from cycle 1 perfectly lines up with pixel 5 million from cycle 95, especially at 80 nanometer resolution?
Another clever solution. Standard practice often uses nuclear stains like DAPI, but those can fade or wash out, needing restaining, which adds variability and cost. PathoPlex uses something different. N-hydroxysuccinimide ester, or NHSE.
NHSE, what does that do?
It's a pan-protein label, so instead of just staining the nucleus, it attaches to proteins pretty much everywhere in the tissue. And here's the key. You apply it just once, right at the very beginning, and it stays incredibly stable through all 95 cycles. It acts like a permanent built-in reference grid or a tissue GPS, allowing for really accurate image registration and segmentation across the entire experiment.
Okay, so APTs anchors the tissue, NHSE provides the map coordinates. Before we hit the software, is the hardware super specialized? Do you need a multi-million-dollar custom microscope?
Actually, no. And that's a big plus. PathoPlex is designed to work with standard inverted fluorescence microscopes, either wide field or confocal, the kind many labs already have.
That helps accessibility.
Definitely. And for the liquid handling, doing all those washes and antibody additions for 40 samples over 95 cycles, automatically they came up with a low-cost automation solution. They essentially repurposed a standard 3D printer to act as a precise liquid handling robot.
A 3D printer?
Yeah.
Seriously?
It cut down the hands-on lab time by about 70%. It's this cool combination of high-tech biology and, well, almost garage-level engineering ingenuity.
That's brilliant. Okay, so the hardware side is sorted, robust, and relatively accessible. Now, the data analysis. You mentioned the software, Spatiomic, and its main output isn't just cell counts, but these clusters. Can you break down what a cluster really represents?
Right. So Spatiomic is this open-source Python library built to handle enormous data sets. We're talking potentially hundreds of billions of pixels per experiment. It uses sophisticated pixel-level clustering algorithms, like self-organizing maps and line graph clustering.
Pixel level, not cell level. Why is that distinction so important?
Think about it like this. Cell-level analysis averages everything within the cell boundary. It tells you the cell is, say, stressed. Pixel-level clustering, because it's working at 80 nanometer resolution, can pinpoint where inside the cell that stress is happening and what proteins are involved in that specific spot.
Like finding the specific faulty component inside the machine, not just saying the machine is broken.
Exactly. A cluster might capture, for instance, a signaling protein moving from the cytoplasm into the nucleus only in one corner of the cell, alongside other proteins involved in that pathway, right there. It integrates location, protein identity, and potentially activity state, like phosphorylation, into one signature. That's the cluster, a spatially precise, integrated biological state.
Okay, quick recap before we move on. The PathoPlex system seems to have cracked some major technical hurdles.
All right, key features on the hardware and prep side include high-resolution imaging down to 80 nanometers, massive marker panels, over 140 antibodies tested, APD's coding for rock-solid tissue, anchoring through 95 cycles, NHSE staining as a stable, permanent reference for image alignment, and clever, low-cost automation using a repurposed 3D printer.
And then the software, Spatiomic, analyzes this mountain of pixel data to generate clusters.
Yep. And those clusters aren't just cell types. They're these integrative co-expression patterns capturing subcellular location and signaling states. They're essentially the fingerprints of specific biological activities or stresses.
So you've got this incredibly detailed map. Let's see it in action. How was PathoPlex first used to find something genuinely new and maybe even actionable in a disease? You mentioned kidney disease earlier.
Yes, the first big test case was in an experimental mouse model of a really severe kidney condition called crescentic glomerulonephritis, or CGN. It causes rapid kidney failure.
Nasty stuff. What did PathoPlex reveal?
The data analysis using Spatiomic spat out dozens of these clusters, these biological signatures. But one cluster, designated cluster 21, consistently popped up. It was significantly increased in the mice during acute kidney injury, and even more so as the disease progressed to full-blown CGN.
And what defined cluster 21? What was its molecular fingerprint?
The analysis showed that one of the top contributing proteins to cluster 21 was PJUN. That's the phosphorylated or active form of the JUN protein, which is part of the AP1 transcription factor complex involved in stress responses and cell proliferation.
Okay, so an active stress proliferation signal. But where was this signal happening? That's the key PathoPlex advantage, right? The location.
Precisely. The high-resolution mapping showed cluster 21 wasn't just everywhere. It was primarily located in two specific cell types, the kidney tubule cells and, crucially, the parietal epithelial cells, or PECs.
PICs. Those are the cells that go haywire in CGN, right? They proliferate and form those destructive crescent shapes in the glomerulus.
Exactly. And PathoPlex showed that the frequency of this PJUN-associated cluster 21 specifically increased within the PECs as the disease got worse in the mice. This strongly suggested that JUN activation in PECs was a key driver of crescent formation.
Wow. So the system pinpointed not just a pathway, but the specific cell type and the activity state driving the core pathology. That's a hypothesis you can actually test. Did they validate it?
They did, quite rigorously in 3 steps. First, in vitro, using cell culture, they mimicked the disease process by stimulating mouse PECs to activate and migrate. Then they treated these cells with a specific inhibitor of JNK, the kinase that phosphorylates JUN. The drug is called CC930, or tanzasertib.
And
The inhibitor completely blocked the activation and migration of the PECs. This confirmed that the JNK-JUN pathway was functional critical for the damaging behavior of these cells.
okay, proof of mechanism in a dish. Did it translate to humans?
That was step 2. Cross-species validation. They used PathoPlex on actual human kidney biopsy samples from patients with CGN. And they found PECs that were positive for both PJUN and another marker of activation, CD44.
Yeah.
These activated PJUN-positive PECs were found exclusively within the crescentic lesions in the human samples. It directly linked the cluster 21 signature found in mice to the actual human pathology.
That's a powerful link. So the final piece, could blocking this pathway actually treat the disease in a living animal?
Step 3, in vivo therapy. They tested the JNK inhibitor, CCNE3, in a rat model of CGN. They gave it both preventively before the disease fully developed and therapeutically after it was already established.
And the result?
In both cases, the drug significantly reduced proteinuria, which is a key sign of kidney damage, and it lessened the structural damage in the glomeruli. It even modulated the expression of that activation marker, CD44, in the PECs.
So they could actually target and interrupt the pathological process identified by PathoPlex's cluster 21? That's pretty compelling.
Let's pause for another quick recap on this finding. The concept. PathoPlex identifies clusters that go beyond just naming a cell type. They define specific functional states, like stress or activation patterns, with subcellular precision. The example. In kidney disease, CGN, PathoPlex pinpointed cluster 21, characterized by active PJUN signaling.
And crucially, it located this cluster primarily in parietal epithelial cells, PECs, the key drivers of crescent formation.
Yeah, exactly. This wasn't just correlation. Validation showed blocking JUN activation with a JNK inhibitor stopped PEC misbehavior in vitro. The same PJUN signature was found specifically in human crescentic lesions, and giving the JNK inhibitor to rats with CGN actually reduced kidney damage.
So PathoPlex found an actionable molecular switch driving the disease.
Okay, that's a powerful proof of concept in a specific aggressive disease model. What about applying PathoPlex to something more chronic, complex, and variable, like human diabetic kidney disease, DKD? That affects millions. It's notoriously heterogeneous. How did PathoPlex handle the variability across, 38 different patient biopsies?
Yeah, DKD is a huge challenge. They analyzed over 400 regions from those 38 biopsies, generating, again, a massive amount of data, over 100 billion pixels. From that, Spatiomic identified 18 clusters that showed significantly different abundance levels between the DKD patients and healthy controls.
18 distinct signatures of DKD at the subcellular level. Any specific examples jump out?
One really interesting one was cluster 19. They labeled it metabolic tubular injury.
Okay, what proteins define that cluster, and what does it suggest about the injury process in DKD?
The main contributors to cluster 19 were 2 proteins, AIFM1 and TRPC6. Now, AIFM1 is linked to apoptosis and mitochondrial stress, while TRPC6 is a calcium channel protein.
Calcium mitochondrial stress. Where was this cluster located?
Overwhelmingly in the proximal tubules. These are the workhorse cells of the kidney's filtration system. So seeing AFM1 and TRPC6 co-localizing right there at the pixel level suggests a specific mechanism. Calcium-mediated mitochondrial stress happening within those crucial tubular cells in DKD patients.
That's incredibly specific. It connects the dots from the overall disease state right down to a potential molecular mechanism in a key cell type. Did these newly identified cluster signatures actually correlate with how well the patient's kidneys were functioning overall?
Yes, and this is really important for clinical relevance. They linked the abundance of these PathoPlex-derived clusters directly to the patient's estimated glomerular filtration rate, or eGFR, which is the standard clinical measure of kidney function.
And what did they find?
They found strong correlations. For instance, another cluster, cluster 28, which represented signatures of extracellular matrix, or ECM remodeling, basically, early scarring.
Fibrosis, yeah.
Right. The abundance of that ECM cluster showed a significant inverse correlation with eGFR. So, as the subcellular signs of scarring increased, the patient's overall kidney function decreased. It builds a direct bridge from the pixel-level pathology to the patient-level outcome.
That implies these clusters could be really powerful biomarkers. Could the software actually tell the difference between DKD and control samples just based on the cluster data alone?
They tested that using computational methods like unsupervised analysis. And yes, just using the relative amounts of the different pathoplex clusters, the algorithms could distinguish between the DKD samples and the control samples with, quote, perfect accuracy.
Perfect accuracy. Okay, that definitely validates the biological significance of these signatures.
Yeah.
Now, you mentioned they also did something called drugability profiling, using the disease signature to find potential drugs.
Exactly. This is where it gets really exciting for potential therapies. They took a list of proteins that were major contributors to the DKD-specific clusters, this DKD signature, and cross-referenced it against databases of known drug protein interactions like string and CTD.
What did that turn up?
Well, first, it confirmed that existing diabetes drugs, including the important SGLT2 inhibitors, do indeed interact with proteins within this DKD signature, which makes sense.
Reassuring.
But more interestingly, it flagged potential new therapeutic avenues. For example, it highlighted PDE5 inhibitors.
PDE5 inhibitors. Aren't those typically used for things like erectile dysfunction or pulmonary hypertension?
They are. But the analysis showed that the PDE5 pathway interacts with that TRPC6 calcium mitochondrial stress pathway we saw in cluster 19. This suggests that PDE5 inhibitors might be repurposed to specifically target that calcium-mediated tubular injury mechanism in DKD. It's a novel therapeutic hypothesis generated directly from the PathoPlex data.
That's fascinating, using the detailed map to find unexpected roads for treatment.
Okay.
Let's talk about the ultimate goal for this kind of tech, catching disease really early, before it even shows up on a standard pathology report. Did PathoPlex look at patients with type 2 diabetes who technically didn't have kidney disease yet, based on histology?
Yes, this was a critical application. They used the full 142 marker panel at 80mm resolution on biopsies from T2D patients who had no overt signs of DKD, according to traditional microscopic examination.
Could PathoPlex see anything amiss?
It absolutely could. Even though the tissue looked normal by standard criteria, PathoPlex revealed subtle but significant changes. It found a decrease in clusters associated with healthy proximal tubule structures, things like the integrity of the brush border, mitochondrial health markers, and lactate transport proteins.
So signs of early vulnerability or dysfunction in those key cells.
Exactly. And conversely, it saw an increase in other clusters, like ones representing stromal cell filopodia, tiny cellular extensions potentially involved in early tissue remodeling, and changes in vascular smooth muscle cells. These are whispers of pathology at the subcellular level.
The disease is simmering below the surface, visible only at this resolution. What happened when these early-stage patients were treated with SGLT2 inhibitors, a standard diabetes medication known to protect the kidneys?
PathoPlex provided a really granular readout of the drug's effects. The treatment did lead to a partial reversal of some of these early stress clusters. For instance, it helped normalize mitochondrial integrity signals in the proximal tubules and seemed to improve the health of the tiny peritubular capillaries.
So the drug was working at the molecular level PathoPlex could see.
Yes, but, and this is important, the treatment did not fully reverse all the T2D-associated changes that PathoPlex detected. It suggests that even with current best therapies, some level of cellular stress might persist.
Which implies there's room for improvement, maybe needing combination therapies or different targets to fully halt the progression, even at these very early stages.
Precisely. It highlights how PathoPlex can offer a much more sensitive measure of both early disease states and the true, deep efficacy of interventions, potentially guiding development of better treatment strategies.
It really unlocks features of injury that are just invisible to classical methods.
Fundamentally, PathoPlex feels like a shift in perspective. We're moving beyond just classifying cells based on their general appearance or a few markers towards a really data-rich approach that captures these distinctive biological signatures across different spatial scales from the whole tissue down to inside organelles. It really does pave the way for what you might call next-generation pathology atlases. And the accessibility aspects, like working with standard microscopes and the 3D printer automation, help democratize this powerful approach.
It's an incredible leap in what we can visualize, which leads to a pretty big final thought for you, our listeners. If PathoPlex can find these distinct, subtle signatures of cellular stress, like that calcium channel activity shifting in just one part of one tubule cell before standard pathology even raises a flag, it makes you wonder, what other complex diseases are we currently missing the boat on? Think about Alzheimer's, maybe early autoimmune conditions, even some cancers. Could their earliest, most critical, most actionable signs be hidden right now at this pixel level? We finally have the lens to look. So the question becomes, what specific subcellular cluster, what hidden molecular conversations should we be trying to decode next to fundamentally change how we diagnose and treat disease? Something to ponder. Thanks for joining us on this deep dive.